University student. Today I want to talk about analysis of KNST cells characterization. Here is the outline of my presentation. This is what I will be presenting today. KNST derived from mesenchymal stem cells. KNST differentiation from seeds. These cells have various differentiation ability, for example, chondrocyte, osteocyte, adipocyte, myocyte, and cardiomyocyte. And these cells have alpha-gal activity. Fabry disease, a deficiency of the alpha-gal due to mutation causes glycolipids known as GL3 to accumulate within the blood vessels, other tissues, and organs. This accumulation leads to an impairment of their proper functions. The present treatment way is enzyme replacement therapy. They must give an intravenous drip with alpha-gal activities first 2 weeks. So we would like to recover an organ by transplantation KNST cells in the organ. So ideal result is patient's burden decrease because it isn't necessary to give an intravenous drip. So process of KNST cells applying clinical for human. First, characterization of KNST cells. Second, transplantation to mouse. Third, clinical trial. Final, clinical application for human. So my experiment's purpose is characterization of KNST cells. This is my experimental purposes: characterization of KNST cells, growth curve, immunohistochemistry of IPGL and subcutaneously implanted sites, gene expression by qPCR, cell surface marker analysis by flow cytic analysis, and gene chip and clustering analysis, and ability of feeder for iPSCs. First, I want to talk about growth curve. This result shows writing a growth curve for each cell per lot type. Number 2 and 3, I seeding cells 0.2 かけの0.2かけの10の6乗を15センチディッシュ1枚に巻きました。で、
Oh, okay, I understand.
ポピュレーションダブリングが3の時に最大2の31.5乗になりました。
はい。
はい。で、はい。で、This shows that I compare the size of cells by passage number. When passage number is young, the size of the cell is small. As passage, the size of cells becomes big. Next, immunohistochemistry subcutaneously implanted sites. This result shows immunohistochemistry. So KNST cells are not differentiation, so it has not characteristics. This result shows immunohistochemistry of IPGL by H staining. So I found that it has no characteristics too. And this is Bemenchen staining, so I found out it has no characteristics too. This result shows gene expression by qPCR, passage 1 and 6, adiponectin, and Fly1 proto-oncogene becomes down, and aglycan becomes high. Passage 6 and 11, Fly1 is not, Fly1 is not expression, so I'm using a graph CDH5. 角フェニ5 and aggregan also becomes high. This result shows cell surface marker per passage number CD19 and 105 is adhesive marker in mesenchymal stem cells. Every passage number says expression. This result shows gene chip analysis. Alpha-gal activity is higher than the others. And Wind 5 and NKX25 of lateral extraembryonic mesoderm marker is high expression. And PDGFR beta of mesenchymal stem cells marker is high, and chondrogenic marker of passage 1 is high, but as passage expression becomes low. And passage 1 and 6 and 11 is mine, and passage 12 by Marie. Passage 1 and 6 and 11 are approximately gathered every group. The characteristic is changing into every cell in the same way by passage. This result shows adipocyte bone cartilage analysis. The characteristics is changing into every cell in the same way by passage 2, and this result shows B cell cytokine heart cardiomyocyte analysis. This result shows the same way by passage 2. And this result shows mesenchymal neuron and stem cells analysis is same way by passage 2. And next, this result shows that I use these cells in the feeder when I cultured Ando cells because it can't grow up well when I seeded seeds by the same density as MEF, so I seed seeds by the half density as it. このメフと同じ濃度で巻くと輪郭が結構ぼやけてしまうんですけど、メフの半分の濃度で巻くと輪郭はきれいになるということが。
Which growth factor do you use? Which medium, growth medium?
全部イプセルです。はい。
Actinoid. I guess this is not good. Not good. Yes. How about this? You don't know. You cannot judge.
Maybe good.
Okay.
でも本当のそのメフで培養した場合ほど表面は滑らかにはあまり見えなかったです。
This is perfect, right? Okay. But maybe how about the detail appearance? Detail.
もっと大きな写真。
Seemingly extend over time. Because it's tiny now. It's like
Is it too thick?
too thick.
Too thick. Is it?
Yeah,
is it not good?
I think just timing
Okay.
to
Okay.
see.
Anyway, good job.
はい。
Great job.
Summary. Growth curve shows KNS disease increases up to 2の31.5兆倍。KNST cells have activity of alpha-gal using treatment of Fabry disease. Even if a lot number of cells were different, it has a similar characterization each passage number. This is all. Thank you.
Thank you. Thank you. Question comment. You have a high power view of the previous slide. Yes, this one. High power view of this slide.
ちょっと待っていただいて。
Yeah, sure. Take your time. you go here?
あ
Where is this?
ネットから取ってきました。はい。あとこれです。はい。
Okay. Yeah, not sure. Maybe not so perfect.
The letter is passed tiny to passage. So that's why the cells seem piling up.
Okay. I'm not sure. I'm not. I do not have expertise on this field, but the margin of the colony is very clear. At the same time, I found certain nuclei and cytoplasm seems to be okay to me, but I'm not sure. I think to me, maybe to
Best way
Yes.
is we need to passage 3 times.
Okay.
Then after that, still good shape is good.
Okay. Thank you for the suggestion.
Right.
Please do that.
Yeah.
Of course, we have feeder-free cultivation of the iPS cell and the human embryonic stem cell right now developed by Hidenori. But yeah, we will see. That's gonna be fun. All right. It is irradiated.
Ah, yes.
You are
そう
right.
です。Yes.
Right.
はい
Question comment.
How do you make KNST cells? Also, how long days?
あ、ですね。だいたい100日ぐらいでパッセージがだいたい13ぐらいになるんですけど、13を超えるともう全然増えなくなります。今のところ。だいたい100日を超えると今のところ増えなくなります。
Could I see? Yes.
これはその増殖曲線が、上にこう平行になったところで止めて見せてるんですけども、本当はここからはもっとこう下がっていきます。で、その最初もらったいただいた14と23と25はその発生経大のその期間が短くてこうなってしまったんですけど、その2と3新しくもらってからその経大するときに1枚あたり 0.2 で薄く巻いてからいっぱいにするようにしたらこういう増殖曲線がもっと増えるような増殖曲線が描けるようになったので、もうちょっとその日数っていうのはこれから伸びるかもしれないです。
Which growth management did you use for this to get to investigate the growth curve? Growth medium.
グロスメディウム、
Right.
はい、これは、アルファメムにその FBS10% 加えたものです。
10%.
はい、20% だと結構最初すごいスピードで増えてしまったので 10% でやるようにしました。
How do you make KNST cells?
町田さんがその ES 細胞をその漢分化させたもので、その下のフィーダーの細胞と上のその浮いてる細胞があるんですけど、その下の細胞を剥がしたものが間質で、上の細胞は今そのまなみさんに移植してもらったりして特性解析をやっています。
So we have 5 different lots
Yes.
for the KNST.
はい、
Number 2, 3, 14, 23, 25.
Yes.
How come you get a lower growth rate in 14, 23, and 24. I'm sorry, 25. Can you tell difference between these two and the
あ、違いですか。そのやっぱり携帯するときに最初ちょっとどのぐらいで増えるかがあんまりつかめなくて、結構濃いめで巻いちゃって、そのもう携帯するのに3日とか4日ペースでやったら下も低くなって、上は結構1週間とかちょっと10日とか最初薄めに本当に巻いて、そこから増えるようにしたら上のこういう伸びが大きくなるカーブが描けるようになりました。
And how about the split ratio? In this case, split ratio.
スプリットレーション。
何分の1で携帯するの？
何分の1は結構小さいです。その1枚あたりがだいたい14取れるんですけど、それをその 0.2 しか取らない。
14ってなんだろう？
14×10 の6乗ぐらい15センチディッシュで取れるんです。でそれで
So you work on a 15 centimeter dish.
Yes.
Okay. And so you get 10 to the 6, how many? 13, 16?
15センチディッシュが何平方
で、いくつ
？15センチディッシュでだいたい1枚 14× の10の6乗コンフレになった時になるんですけど、だいたい15センチ1枚で 14× の10の14×10 の6乗になるんですよ。でそれを 0.2 ずつ巻いてるので
0.2
そうです
ってことは14分の 0.2。
そうです。なので14を全部 0.2 ずつ巻いたらすごい膨大な数になっちゃうので、その 0.2 を数枚巻いて、あとはもうペレットにするとかして取ってあげます。はい、
14分の 0.2 っていうことは、100分の1弱だから64分の1分。So the 64 means 2 times 2 to the 2×4 2、4、8、16、32、64 2 to the 6th, so 2 to the 6th. In that, there's two passage in so every week you do the passage.
Yes.
And every passage cell grows at 64-fold, and that means 2 to the 6. So thinking of the population doubling, it could be like a 6. So every day it could be one division, right? Every day, 6 days, 6 days, yes. Good growth rate. Very fast, right? とても早く増殖するますか？
巻くときに濃いとすごい早く増えます。細胞が若いほどすごく早く
増えます。0.2×10 の6乗を15センチに巻くって、スカスカだったのが突然100倍ぶわったから1週間で。
だいたい 1,2 週間ぐらいでいっぱいになります。そのやっぱ経来数が若いほどその速さがすごい速くなる。
So that in 14 days, that means in 2 weeks, it goes up like 1, 2, 3. That means 10 to the third. That means 1,000-fold in 14 days. Again, I would like to calculate 2、4、8、16、32、64、128、250何歩の500の1000、10の10乗。So in 2 weeks, the population doubling increased like 10. Okay, by 10.
そうですね。
1, 2, 3, right? 1, 2, 3, and more, maybe more. Good. How about the flow cytometric analysis? You have a result?
これです。はい。そのパッセージ1、5、11で若い普通、置いているとして、今90と105どっちも見たんですけど、どっちも発現している。
How about the other markers?
CD90 と105、寛容系に出る。
My question is, have you investigated other markers? Like CD 29, 44.
とりあえずこのラボにある平さんのものをお借りしてやって買うかどうかはとりあえず平さんのものをやってっておっしゃったので買うかどうかはまた相談させていただきます。
Very good idea to save money. Right. Yeah, thank you.
You should separate give time. Actually, we have standard markers. It's not only two. It's the world standard markers, 5 or 6. They have.
Question?
So, to examine growth curve, you use 150 millimeter dish. I recommend you should use more smaller dish because cells is very scant at the beginning point. It's not good. Maybe if when you use a smaller dish, 160 millimeter dish, the result is much better. Probably related to atrophic factor. Cells environment is very good.
How sparse it is? どのぐらいスカスカ
巻いた時そんなスカスカではない。
2 times 10 to the fourth, right? No. 2 times 10 to the fourth, right? No. It's 2 times 10 to the fifth. 20万最後。
0.5かけの10の5乗。
もう一回日本語。
0.2かけの10の6乗を今15センチに巻いてる。
That means 2 times 10 to the fifth. That means 200,000. Yes. But that's enough. I think so. Right? Question?
Let's see clustering data.
これがその全部
So the KNST, you never done differentiation KNST cells. So KNST cells, this means express these adipose calcularia also neurons marker expressed.
分化リードしてるか。
分化してないのにそういう脂肪とかのマーカーが発現してるのって。
発現している。も もし仮に発現していたとしてもすごくその小さくだと思います。それで分類分けしてるんだと思います。これで例えば脂肪とかなんですけど、全然2とか3とか、大きくて5なので全然発現してなくて、その中でこう分類分けしたんだと。
So maybe she did not gene filter in classic.
No. She did. She did. Actually, Yoriko did. And Yoriko filtered the gene based on the represented markers, genes, for each differentiation phenotype. And of course, at the very beginning, she filtered very low level of expression. And after that, she classified all the gene markers. So, she didn't use these genes for the hierarchical clustering analysis. Never. Please, yes. Don't worry. Yeah. This. All right. So the important thing is that could I see a hierarchical clustering analysis again, please? Hierarchical clustering analysis. Right. Maybe we need to elucidate which gene could be changed during the passage, because this kind of experiment is essential only when these cells are different. If they are the same, such analysis is boring, especially like this. Yeah. This, right? Okay. Question. Comment. 町田さん。No, he's not here. Okay. Thank you. And you.
Thank you so much.
We have 2 U's.
Yes.
You.
The other U. I call him you.
なんか、池本さんにこれが使えない。
I call Ando you and you. Just you. Perfect. So, Hajime, you're gonna help him, all right? And Marina, you 3 ladies and gentlemen, a team. Okay.
よろしくお願いします。
そのまま行こうか。
Today talk about medulloblastoma generation in Gorlin syndrome induced pluripotent stem cells. Today's outline. First, I talk about hedgehog signaling pathway and Gorlin syndrome. Hedgehog signaling play key roles in development and disease, such as pattern formation and cell fate decision, cancer, and developmental anomalies. PTCH1 acts as a primary inhibitor of the hedgehog signaling pathway. When the hedgehog ligand binds to PTCH1, the pathway occurs activation, releasing PTCH1 from small inhibition and activating group transcription factors. Gorlin syndrome patients have heterozygous germline PTCH1 mutation. PTCH1 mutation have enhanced hedgehog signaling. As a result, cell fate change and excessive growth in some tissues. and loss of the remaining wild-type allele occurs the tumor genesis. Next, show clinical features about Gorlin syndrome. Gorlin syndrome is characterized by a wide range of developmental abnormalities for example, leaf morphological abnormalities and various tumors, such as Ikemoto, basal cell carcinoma, and medulloblastoma. Especially eye focus medulloblastoma. Medulloblastoma is malignant childhood brain tumor. Genomic studies have 4 distinct medulloblastoma subgroups, each with characteristic molecular and clinical features. Sonic hedgehog subgroup medulloblastoma originates from granular neuron precursor. A cell type that is present in the developing cerebellum and whose proliferation is regulated by the sonic hedgehog pathway. With current therapy, 5-year overall survival of the group is approximately 75%, but survival is frequently accompanied by severe morbidity. For patients with recurrent disease, survival is dismal. Therefore, improved therapy is needed not only to augment survival, but also to prevent recurrence and decrease the morbidity associated with current therapy. 治療効果があったとしても、5年生存率が上がったとしても、後遺症がすごく残るので、そういった後遺症が残らないようにな薬が求められているということです。Now, very effective drug approved by the FDA in 2012 for the treatment of locally advanced basal cell carcinoma to prevent recurrence of advanced basal cell carcinoma. The drug name is Bismudeyu. It planned to be used for other tumors, medulloblastoma. Bismudeyu is antagonist of smooth receptor, which is part of inhibitor of the hedgehog signaling pathway. But this drug have many side effects. For example, diffuse alpasia, dysgeusia, weight loss, asthenia, and teratogenic effect. About 40% of patients cannot continue Bismudeyu therapy because of these side effects. My purpose is modeling Gorlin syndrome using iPSL for clarification of pathogenic mechanisms and for screening novel compounds to treat the disease. So I try to establish iPSL from fibroblast. Patient cells were obtained from 4 patients. This slide shows PTCH1 mutation in genomes from patient blood. 3 patients' type of PTCH1 mutation is frame shift, and 1 patient type of PTCH1 mutation is deletion of full. The frame shift mutation will alter the first stop codon encountered in the sequence. For that reason, the polypeptide binding created could be abnormally short and will most likely not be functional. This slide shows a flowchart of generation of iPS cell from Cori syndrome-derived fibroblasts by Sendai wheels. When iPS cell colonies are ready for transfer, pick up and transfer undifferentiated iPS cell onto fresh milk culture dish for expansion. I successfully generated iPS cell from human cells with a mutation in the PTCH1 gene. Next, I performed immunocytochemistry of pluripotent cell-specific marker demonstrated expression of SSEA4, TRA160, SOX2, NANO, OPTO34. Next, I analyzed whether the Golden iPS cells have the competence to differentiate into specific tissues, demonstrate the three primary germ layer. Next, I performed karyotypic analysis. The three Golden iPS cell clones had an intact karyotype. Next, I analyzed PTCH1 gene and expression of hedgehog-related gene. This shows that G72 iPS cells genomic analysis of PTCH1 gene. In G72 lymphocyte, a deletion of G at nucleotide position 272 of the PTCH gene was detected by sequence. Surprisingly, G72 number 1 iPS cell has a different mutation site in the PTCH1 gene. A deletion of G at nucleotide position 272 of the PTCH1 gene was not detected, but a deletion of G at nucleotide position 272 of the PTCH1 gene was detected.親細胞と樹立した iPS 細胞でゲノムの変異の位置が変わっていたという結果が出ました。So I performed genomic analysis of PTCH1 gene in another cell line. As a result, G72 iPS cell number 1 and 5 has a different mutation site in the PTCH1 gene. Number 2 has the same mutation site in the PTCH1 gene. G72 iPS number 1 detected to be derived from patient's cell from the result of STR analysis. Therefore, there is a possibility that mutation site change during reprogramming process. Next show, I assess hedgehog signaling by means of qPCR analysis of the level of messenger RNA encoding the PTCH1 and the hedgehog target gene GRI1. Look at the PTCH1 expression level. Golden iPS cell expressed lower than EOM iPS cell. On the other hand, hedgehog target gene GRI1 expression were not higher in Golden iPS cell than in the EDOM. And next, I compare of the cell proliferation speed because hedgehog signaling pathway promotes cell proliferation. Nevertheless, Gorin iPS cell and Eden were same cell proliferation speed. Although PTCH1 mRNA were lower in Gorin iPS cell than Eden iPS. Gorin iPS cell could not be confirmed change in hedgehog signaling response. Next, whether or not malignancy cancer were observed in any of the Gorin syndrome teratoma. Histological analysis of demonstrates that 3 germ layers were generated as shown by the presence of ectoderm, mesoderm, endoderm tissues in the teratoma, implying these cells had potential for multi-lineage differentiation in vivo. And medulloblastoma was detected in all the teratoma generated by all iPS clones from 4 different patients.でこれは、那須さんにも見ていただいて、北里の病理の先生にも見ていただいて、一応髄芽腫ではないかという所見をいただきました。Next, I perform immunohistochemical analysis of medulloblastoma. Ki67 is marker of proliferation. This is positive. SFRP is characteristic of sonic hedgehog medulloblastoma marker, but negative. New N and GFAP is neural antibody. But negative. Other region teratoma,ポチコンでは、他の、テラトーマの領域では染まっている箇所があったので、抗体に問題はないと思われます。で、So I will try to another neural antibody and sonic hedgehog medulloblastoma marker, GRI1. Next, I perform laser microdissection for isolating medulloblastoma. Whether LOH occurred. There are several mechanisms which can cause cell to lose its normal gene and thus be predisposed to develop into a tumor. They may result in a LOH. I pick up medulloblastoma and cartilage. This is control. This slide shows the G11 medulloblastoma cells genomic analysis of PTCH1 gene. This sequence is near the region containing the mutation in one allele. In G11 control, a duplication of GC at nucleotide position 3130, 3131 of the PTCH1 gene was detected by sequence. Medulloblastoma cell of mutation site were not changed in the region. but this sequence is only near the region containing the mutation in one allele. So another site possibility mutation. Summary. iPS cell from each Gorlin syndrome cell was successfully generated. Heterozygous germline PTCH1 mutation was suggested not require pluripotency. Surprisingly, the parental cell of G72 iPS cell has a different mutation site in the PTCH1 gene. There is a possibility that mutation site change during the reprogramming process. Successfully generated medulloblastoma model with iPS cell derived from patients with Gorlin syndrome. The Gorlin iPS cell model serves pharmaceutical approach to find low molecular to treat medulloblastoma and Gorlin syndrome. That's all. Thank you.
Thank you. Question, comment?
So how many clones G72 iPS have you checked the mutation?
確認したのは3クローンです。そのうちの一つが親株と一緒の変異を持っていて、他の2株が変わってきました。
はじめ。
Yes.
Could you explain more about this, please?
理由ですか
Right. I think this is contamination possible. But as you mentioned, this is a rare mutation. So rare, and this is the first mutation ever reported, right?
Yes.
First, and it's a frame shift mutation, and so this is quite reasonable for the Gorlin syndrome. So this is a big enigma. How to say? How do you think about that? Or how about how do you think? Have you done the short tandem repeat analysis for these 4 clones, control and 2 and 3, right? This one, and also you have another one, right? The other one. So have you done the STR analysis?
1番のみしかやってないですね。1番です。
So it's
これは
the same.
変異部位が変わっていたクローンで、
Probably. To me, there must be a contamination during the DNA preparation. Otherwise, I cannot tell why this happens. Suggestion? At this moment, I don't know. But If it's true, if a short tandem repeat and the mutation from the same genome, maybe we need to think of the mechanism of to have a new mutation in the same gene during the IPS generation. That is not very rare, but I understand Toshiyuki denied the contamination of the samples. Toshiyuki is a president of the Kitasato University School of Medicine right now. He got promoted last month, right? But I still don't know. I'm sorry, I don't believe this.
So is mutation reproducible? So how about try the same experiment?
Yes.
Is not,
So the new label of the sample is different
もう一回やって
という長尾さん
リプログラミングもう一回する。でもう一回同じミューテーションをもらって、
はい
この際にもらって。
I do not agree with
もらった
him.
のは千葉大からもらってるんで
もう一回。
He wanted to reproduce the generation of the new mutation site in the during the reprogramming, but I do not want to do that. I don't want to do that because it's not a central theme of our study. Our central theme is that explain please, Hajime. Our goal for this generation of the IPS in the Gorn syndrome is
薬を作るという
right. So for to do the pharmacology, what do we need to do? We need a biomarker, right? Of course, neuroblastoma is great, but to screen low molecules, this is too difficult to analyze every time in vivo. Medulloblastoma by histology, it's a big, it takes a long time. So it could be a final, but it's not to find the screen low molecules. So probably we need a biomarker. Please concentrate on that, please. All right?
Yeah.
All right? Please. Marina, please. So, to you don't know, what can you do? What can we do for this?
today
No, in the future, near future,
future
next month. What do you do for this experiment? Maybe perform the sequencing again. Ask Nagao-san to do this again. How many members believe this? This gonna be true.
じゃあ信じて。
I like it. I like it. Uda-san, I like it. Atsushi. Good guy. How about Atsushi? Okay, nice son. You're a friend, right? Classmate. For 6 years. Right?
6 years
Huh? Alright.
ちょっと日本語にする。ブラストマーのレーザーマイクロダイセクションのやつなんだけど、結局2ヒットではないのにブラストメアができ
ツーヒットかはまだわからない。
でもそれ見ると2ヒットにそう他の領域があって
他の領域の可能性も
それは突然 iPS になって他の領域が入るっていうのは、一つのケースとしてあったんだろうけど、これレーザーマイクロダイセクションで取ってきたってことはかなり微量だよね。それをガンガン PCR かけるってことは、ちょっとしたコンタミネーションでもうバイアレリックになるはずだけど、 だからその PCR サイクルをどれぐらいにしてるかっていうのはアレリックエクスプレッションとかを見るときには結構シビアで、例えばインプリットジーンでも PCR ガンガン回せばバイアレリックになってしまうので、そこをやるんであれば例えば定量性を持たせるんだったらパイロシークエンスとかしてでどっちの案件がどれぐらいのパーセンテージに入ってるかっていうのができるの、そういうシークエンスを。そうするともしかしたらコンタミネーションで本当は2ヒット入ってるのにもう一つのやつがガンガン増えてきちゃって結果検出してるっていうのもあるし、あるいはレーザーマイクロダイセッションでも本当にもう真ん中の部分だけを取ってくるとか、そういうふうにしたりして、いわゆるそのアーティファクトを消すっていうのをやった方がいいんだよ。
これ PCR かけてないよな。ダイレクトシークエンスだね、普通に。
でダイレクトシークエンスで PCR かけないと当然読めないんですよ。
だってゲノム持ってきてプライマー用意して
ファンがプライマーでそう、いや多分一回増やしてると思います。
増やしてる
増やしてます。
それじゃダメだよね。
多分普通にシングルいわゆる普通のシーケンスプライマーだけでっていうのはちょっと走らないんじゃないですかね。
そしたらね、CDN 取れないか困ったね。
シーケンス
あそこの壁のアイセルは CDN 取ってんだよね。それでロスオブヘッドのサイクロシティを証明してるので、なんだじゃあこれを福田さんのおっしゃってる可能性の方が高い。さっきからこのピークの高さに非常に懸念を示していて、
それサンガーのやつってあまり僕の経験的にあれは定量性がないっていう印象を持ってるんですけど、
だけど定量性がないかもしれないけれども、そこにはプレフェレンスっていうか癖があるの、機械の癖で、機械の癖でもよく見てもこの他の高さはほとんど一緒なのに、ここだけ高さがずれていて、まして部分的になんか高さが違うなという印象を持っていたんですね。これどっちがもうあまり最近現場にいない人は分からない。赤がどっち？緑は何？
が T ですね。
はい？
T ですね。
赤が T。赤が T。赤が T で上がってる感じに見えるのかな、こっちのあれ。
パイロシーケンス、そういう定量ができるシーケンスが
そうか。ごめんなさい。PCR はかけてないんだと思うんです。なんか最近はもう感度がいいから。いやいや全然パイロシークエンスはどういうメカニズム？
色素をそれぞれ違うのを取り込ませてプライマーをそれであれシーケンス PCR ってプライマー一つじゃないですか。それをそれぞれ違う方向から出したらまずで色素を違うのを入れて確か検出するんだ。確かそれで定量ができるっていうのをちょっと詳しく原理は忘れてしまったんですけど、中林先生とか知ってるんですよ詳しく。
DNA の濃度ってどれくらい必要なんですか？
それは結構必要だと思う。だけど PCR コードシーケンス PCR をかける前に一回増やさないといけないから。
最初のそのスタートのタイムをシングルステージから始めたらもうそんな時間がない。
いやそんな時間がないというかですね、そんな能力がないようちにシングルセルからシークエンスするって。
いやいやそうじゃないです。最初の iPS 化するところの最初のところを、シングルセルというか、こう概要書していって最初の時点を一つにしてしまうので、
いや細胞無理でしょ。そんなんできないでしょ。要するにね、サブクローニングしようって。それでサブクローニング10個取ればこんだけの頻度であれば、でもないよね。なぜかというと元々のやつにこの新しいミューテーションの影すらないんだろうね。
そうですね。
ない影すらない。何言いたいかというと、could I see your sequencing of the parent as well? Yeah, right. This one, parental cell.
やりたいですね。あ、これですね。
Right. So Can you tell, is there a possibility of the new mutation here? I'm not sure. Have you checked here?
T です
T. G, G. Very complicated.
この
トリプル G.
G が抜けて T に変わってるのが親のシークエンス。
C, T, G, G, G. Ne? 得意じゃない。C, T, G, G, G. This is deletion side, right?
上は
Deleted.
そうそうそう。
Deleted
はい
nucleotide.
はい
And then T, T, G, T, T, A. So, let's see. So, maybe in this cell, this could be a heterogeneous, these parentals may have the heterogeneous mutation site. If that are the cases, we see T and G, right?
はい
T and G,
は
right?
い
And in this case, T only T, and T and G, right? T and G,
はい
and then T and G. Very confusing. I'm not sure. Please make sure if there is what type of the nucleotide curve could be obtained from the assumptions of the Kenji's. All right. Based on the Kenji's assumption, there could be different type of the these curves. So please check it. Yes. これが3つのタイプのゲノムであれ3つのゲノムのタイプがあるわけだよね。一番多いタイプはどういうタイ
一番多
プ？
い
予想されるケンジ宮田さんがおっしゃってるようなことがあればどうなるか。
これが多いとは思う。リプログラミングされたのはこれがたまたま多かったんですけど、これがやっぱり多いな。
一番多いのは正常タイプだよね。
あ
なぜかというと、
はい
50% がそうだから。残りが2個に1個だから半分半分の 25%、25% だとすると、どういうカーブが想定されるかを自分で手で書いていくの。3通り選ぶよ。リンパ球がゲノムは2コピーあるでしょ。2コピーの中でこれが50、正常が50、ミューテーションタイプ1、メイテーションタイプ2、それが50、25、25の割合で入ってるとして、カーブが大体どうなるかって想定されるでしょ。それで分かっちゃうから。いくら定量性がないと言っても、定量性がなくなる方向に、なくなるというか、ちょっとでも入ってればそこが、増幅される方向になるに決まってるから、宮戸さんの想定はこのカーブから分かるので、このカーブから分かる、ここで見れば分かるので、それをこうなるはずだっていうのを見てそうなってるかどうかを見ると、3通りあるのか2通りしかないのかを見てみない。コロニーは取っちゃダメ。
はい
タックマンのスニッププローブ使ってみたら、1塩基の違いを検出できるはず。そうすると違うコロニーで取ってきても何コピー入ってるかっていうのが計算できる。そうです。iPS。
But the point is that contamination, not that. No, we are focusing at that. I understand. Thank you. I really thank Atsushi for believing this brilliant bet. But probably what we need is to eliminate the possibility of the contamination of the genome, not the cell. Genome. 僕が想定してるのは細胞段階のコンタミじゃない。ゲノム段階のコンタミだから、いくら小タンデムリピートとゲノムがちゃんとあったからって言っても、 こっちはだからゲノムを業者に渡す時と長尾さんに渡す時でちゃんと同じものを渡してるわけだけど、業者はちゃんとやったけど長尾さんは別の検体とずれたら当然同じ検体から STR は一緒だけどもミューテーションの位置は違うっていう結論が出てくるんで、それ長尾さんだっていやそんなコンタミなんかありえないって言うけどものすごい数やってんだからだから長尾さんから STR を出させんなら分かる、その同じ検体でなら分かるけど、君からこう出してるからここで検体間違いが想定されるので、じゃあなんでレアケースのやつがありえるんだと、これ僕分かんない。なんでレアケースが
そっちにミューテーションが入ってるゴーリンの患者も
いやいないんです
いないんだ。
そういないんだ。日本人少なくとも世界でいないんだよね。
多分ね
世界でだから初めてのミューテーションが iPS で見つかってきちゃうんだ。それも iPS 化の過程で。
それでもコンタミだとすごい確率ですよね。そこにミューテーションが入った DNA が入る。
コンタミどこコンタミだったら別の患者から長尾さんが持ってるやつが偶然世界初の検体だ。だから僕は淳と同じ意見持ってるわけ。ありえない。ありえない。なぜかというと患者さんからもらったやつと別の位置にミューテーションがプログラミングの過程で起こったっていうことは一回治ってまたそこに入ったとか、または同時に入ったとか、片一方入って後からのもう残りは治った。無茶だ。普通ありえない。そんなの同じ場所、それもパチワンだけって理論的にはありえない。これゴーリーの人は純粋に信じてくれるかもしれないけども、一般的にはちょっと違うな。多分長尾さんの混入と、あとは初めての検体ミスと初めてのゴーリンのミューテーションが見つかったんだと思うんだよね。ただ調べるべきは何人ゴーリンの検体が北里にあるか。
100検体以上ある。
100検体ってことは十二分にある。僕てっきり5検体とか10検体かなと思ったんだけど、だから千葉大学から来た4検体ぐらいしかゲノムの解析してないのかなと思ったんだ
100件って全部シーケンスしかいないな。それだら出てくるかもしれない。
けど、ホットスポット
多分
そうない
ないと思います。
ないってことはね、ってことはやっぱり世界初だらけなんでしょ、ミューテーションの位置って。
そうですね。
ね
親子ぐらい多分同じやつはないかもしれない。
はい、
親子ぐらいしか多分同じやつはないかもしれない。
じゃあ世界初って言っても僕らがよく知ってる病気のファウンダーっていないんだ。ファウンダーっていうのは小児科日本人の中で1人目がいて、それから1人がいてずっと全員日本人全体の患者は全部そこの人から始まったっていう。いないの。じゃあバラバラなの。ファウンダーいないの。
バラバラにいます。バラバラに。
ミューテーションのサイト
はい
ね。
みんな症状違う。
みんなちょうど一緒ですよ。大体
症状は
ラージデリーションとかでその別の領域まで行っちゃうと非特異的な症状が出るらしいんですけど、基本的には一緒。
まあ一緒というか多分バイアスかかってて症状から診断してミューテーション見てるので、ミューテーションから症状っていうのは多分ないので、ちょっとそこはバイアスきっとあると思うんですね。ミューテーションあっても症状出てない人いるかもしれないので、それはちょっと何とも言えないと思うんですね。そこはちょっと症状は同じなのは当たり前ですよね。症状同じ人を集めてるので。
多分そこら辺で整理つくと思うんだ。100あるんだったら検体間違いないって言うんだけど、そのゲノム、長尾さんが調べたっていうゲノムでスニップやってSTR そのゲノムくれますかって言ってちょびっとくれ、それで STR やってうちからもう一回出すんじゃなくて、うちからもう一回出してね、 だって2度目のこのためにはまた別のミューテーションが出てくるかもしれない。
この iPS お前が作ったんでしょ。
そうですね。
じゃあこれお前がシークエンスすれば違う結果になるかもしれないけど、シークエンス向こうにやってもらったんでしょ。
そうです。
あその通りだよ。
そうすれば1個しかないんじゃない？
1個しかないんだよ。そうだ。それが一番早いわ。
プライマーだけ教えてもらって入れるだけ、こっちで普通に
あ、本当にそう。
ファスナーで頼めば。
何バカなこと言ってたんだ。それ一番いいわ。よかったね。解決しましたね。うちでやりゃいいんだよ。あとお前それ考えろよ、それ。考えて自分でやりゃいいじゃんって。すぐ解決するじゃん。自分で持ってる iPS から2個やって、だけど不思議なのに2個この試するってあるか。なるほど。Okay. Now we have good suggestions. Thank you. Thank you so much. So one more talk for me. Ando, you mentioned about the KNST. I've been working on the KNST for 30 years. Believe it or not. So I just like to show what I did. I have been done in my life. Yes. Alright. So this is our symbol. Then remember, further, murder, patient with some pains, baby, alright? And so, or doctor, nurse, patient. Finally, society, and family, and kid. So, maybe it's our symbol. So to get KNST, I have been working mainly from bone marrow. At the same time, I used a fetus under the approval of the IRB, Keio University School of Medicine. So, right now, today, and you mentioned that she got KNST from the human embryonic stem cell. Let's see. So, from the bone marrow, there are KNST yellow, showing yellow. This is vein, arteries. At the same time, we have KNST. We call it marrow stroma cell. So it's a MSC. We call it MSC in 1991. At that time, we do not have the term mesenchymal stem cell. At that time, we used marrow stroma cell, MS as an MSC, designated MSC. But at the same time, in 1991, Pittenger reported paper in Science of the mesenchymal stem cell. But I also reported the multipotency of the marrow stroma by using the murine marrow stroma cell. And the differentiation capability includes bone cartilage fat, cardiomyocyte, skeletal myocyte, and neurons. So we used certain type of the marrow cells and injected subcutaneously injection of the mice. We see bones, external dense nodules at 2 weeks. Histological analysis revealed this osteogenesis, bone formation, with hematopoiesis. So I wanted to generate the bone with a shape which I want. So for that, I use a scaffold. These scaffold with collagen type 1. Fabricated collagen fabricated scaffold. We call it polylactic glycolic acid sheet with collagen type 1. So we put the cell on it and then fabricate the seed, and then we get a bone with a tube structure. This is an X-ray, and this one is a histological analysis. We see a very clear bone with a tube-like formation. At the same time, lab member wanted to make a nuts, bone nuts. Nuts, see? Of course, we cannot, it's very hard. So it is very hard to, so we cannot change, it's a bone, but a nut. So in the at the subcutaneous position, Tsuchie generated this nut-shaped bone. Finally, we wanted to generate a phalanx-like bone here. This is graduate student's thumb, his thumb, and this is the bone. We call it phalanx. So finally, he generated a phalanx in mice. So at the same time, this was by another graduate student, Sugiki, wanted to generate a cartilage with certain condition. He successfully generated a cartilage, subcutaneous tissues. You see a subcutaneous region, and the matrix stained positive for collagen type 2. So he was happy, and then analyzed he wanted to generate in vitro and in vivo. So these cartilages are very positive, exhibited a metachromacy in vivo. Metachromacy means stain differently. Like usually this is a tourism blue, but at 4 weeks after injection, the cartilage showed metachromacy. So he generated the cartilage, and then finally we found hematopoiesis within the cartilage with the bone formation. So I wanted to move to the neural differentiation from the osteoblast. So by using a demethylation agent, 5-azacitidine, they started to protrude the axes from the cells, which are positive for NUN, GFAP, and HU and TUG1. Today, Ikemoto showed that medulloblastoma were negative for these markers. But I believe they're medulloblastoma. I believe so. No doubt. But using osteoblast, we could show the neural differentiation with a very bright cytoplasm with a long protrusion axis, which are positive for MAP2, a marker for neuron. They are MAP2. So finally, we performed physiological analysis. Patch count, no, this is not patch count. How did I say? I didn't remember. This is, how do I say? I don't remember, with exposure to glutamate, neurotransmitter, at the same time, high potassium, they start to depolarize. That indicating these neurons are functional. So finally, I wanted to do the flow cytometric analysis of these mouse marrow stromal cells. So I generated KUSA A by Kusakari-san generated this. That's why I use this one. Of course, I was in Keio University, so I say it's a school of Keio University School of something. But actually, Kusakari-san did this. So that's why I named this cell name KUSA, A, O, CMG. This is cardiomyocyte. And the 15C, KUMU4, 5. And so what I wanted to tell you is that there are specific markers. For example, maybe under you, maybe I want you to do the endothelial markers. Of course, they are negative because from the G-chip analysis, they do KNST did not express these genes. I checked, and also I want you to analyze CD34 CK scaman. I guess scaman is a CD19. So you did that, all right? So the 140. Did you do that? No, you did CD90 and 105, right?
Yes.
105, right? Right, 105 here. So I'm very happy that the KNST are positive for the 105. This is a receptor for the certain cytokine. I didn't remember it. Maybe TGF beta something, right?
Yeah.
So I want you to analyze this for cytometric analysis. That's why I move. So this is all by neuron cells. So I wanted to move to the human cells. So I obtained H1 from the 91 years old lady, and after under the condition of the, so at that time I wanted to analyze the stress, maybe yes, Chihiro, and right, stress. I wanted to see a stress, but for me, it's a stress. Could be a cell growth is a great stress, and the fast growth is a very stress. So I wanted to see if that. How about the stress? Important thing is that I wanted to. So first, I wanted to analyze the. This is endometrium of the derived KNST. Endometrium. Endometrium is. I'm sorry. It's yes. No, endometrium here. This part. We get we had gynecologist. She moved to the Nara Prefectural University School of Medicine, and then we get endometrial cell, and then we did cardiomyogenic differentiation, and so we get the two cell lines, and we also perform the flow cytometric analysis. CD91 could be positive. See, this is NGF receptor, and also the CD44 could be positive, 55, 59, 90, right? But see, 214 could be slightly positive for the well, positive for the 105 and the 166. I'm wrong. Someone is not a CD90. I don't remember. Someone is something. And these stem cell markers were negative for this mesenchymal cell. So at that night I mentioned these 90, 29, and 44, 59 positive cells could be stromal cell with a multipotential. So, and also I wanted to do the safe, fast, and large volume of cultivation with this marrow stromal cell. At that time, I used the word mesenchymal stem cell. I changed my mind in 1990s. Point what I want to tell you is that the stress is very important to get a large number of cells. What's interesting though, I got published with this with a strong signal. Actually, factor X is a PDGF and I'm sorry, I forgot. PDGF, one more cytokine. Accelerate the cell growth in the mesenchymal stem cell or marrow stromal cell. However, lifespan decreased. So, faster cell growth, it could be a stress for the cell. And so we developed medium X. We call it MF medium, which Chien used for the human iPS generation. He got published the human iPS generation by using medium X. Actually, it's an MF medium, but this MF medium increased stress marker P16 in 4A, and so we use fetal calf serum at the same time. Oh, I remember. It's a basic FGF and PDGF are a potential inducer of potential inducer of P16 in 4A. That's why the same result was reported in genes and development. 1 year, almost the same. So that's why we submitted the same result to the Journal of Cellular Physiology. Important thing is that PDGF, basic FGF increase P16-4A, resulting in a premature senescence of the cultivated cell. Premature senescence. So I wanted to see these cytokines, low molecules, to see if these proliferate capacity at the same time. So what I found is that growth correlation is R is 0.7. So our conclusion is a cytokine which increase the cell proliferation induce P16 in 4A. So final conclusion was serum is not the kind of best at that moment. Of course, serum is not so good because it's undefined, molecularly undefined media. So we prefer to use the growth medium, a growth factor, but they grow rapidly, but induce a premature senescence. So this one, so we call it, at that time, around 2000 to the 2000, I wanted to generate, of course it's a first generation, a second generation was like this. So this is our medium, MF medium. But finally, we wanted to have a third generation or the next generation that proliferate but not inducing the iPS cells. Not inducing P16 in 4A, I'm sorry. actually, this was achieved by right now. So we do not need to do that anymore. But this stress signaling pathway was elucidated by Naoko. Yes. You are mentor. She elucidated these pathways very clearly and with aging power. So important thing is to get a safe fast growth and large volume. To get a large volume, we need to do that. We need to do the more science. ちょっと昨日部室長会議でですね、